Chapter 8 Audio Lecture Genetics, Conception, and Fetal Development. Everyone expects a perfect baby, and if a genetic disorder occurs, the shock and grief can be as if the child died, and really, their perfect baby did sort of die. Understand that they will go through stages of grieving, starting with shock and denial, and they really cannot be pushed through these stages. You have to give them time and help them concentrate on immediate needs and very short-term goals until they are ready to start anticipating the future. According to the Center for Disease Control's, birth defects and genetic disorders occur in 1 in 33 infants born in the United States and causes 1 in 5 infant deaths. Genetics is the study of individual genes and their role in inheritance. Genetics provides the tools to determine the hereditary component of many diseases. It improves our ability to predict susceptibility, onset, progression, and response to treatment. Newborn screening is perhaps the most widely used application of genetics in perinatal and neonatal care. Our ability to diagnose genetic conditions is more advanced than our ability to cure or treat the disorders. However, accurate diagnosis has led to improved treatment and outcomes for those affected with these disorders. Genetic disease affects people of all ages, socioeconomic levels, and racial and ethnic backgrounds. Genetics has been a part of perinatal care for decades. Ultrasounds and maternal serum screenings have become a routine element of prenatal care. Preconception carrier screening for conditions such as Tay-Sachs disease has been in place among high-risk populations such as Ashkenazi Jews. Amniocentesis and chorionic villus sampling are diagnostic tests that may confirm a genetic anomaly in a developing fetus. Genetic disease affects individuals, families, communities, and society as a whole. Advances in genetic testing and genetically based treatments have altered care. Genetic disorders affect people of all ages, from all socioeconomic levels, and from all racial and ethnic backgrounds. They affect not only individuals, but also families, communities, and society. Advances in genetic testing and genetically based treatments have altered the care provided to affected individuals. Improvements in diagnostic capability have resulted in earlier diagnosis and enabled individuals who previously would have died in childhood to survive into adulthood. Some disorders appear more often in ethnic groups. For example, Tay-Sachs disease in Ashkenazi Jews, French Canadians of the eastern St. Lawrence River Valley area of Quebec, Cajuns from Louisiana and the Amish in Pennsylvania, beta thalassemia in Mediterranean, Middle Eastern, Transcaucasus, Central Asian, Indian, and Far Eastern groups, as well as those of African heritage, sickle cell anemia in African Americans, Phenylketonuria or PKU in Irish, Scots, Scandinavians, Icelanders, and Polish, and cystic fibrosis most often seen in Caucasians, Ashkenazi Jews, and Hispanics. Chromosome abnormalities are a major cause of reproductive loss, congenital problems, and gynecologic disorders, and account for approximately 4 to 7% of perinatal deaths and 0.5 to 1% of infants born with multiple anomalies. Several sex chromosome abnormalities are caused by non-disjunction through gametogenesis in either parent. The most common deviation in females is Turner syndrome or monosomy X. The affected female exhibits juvenile external genitalia with underdeveloped ovaries. She is usually short in stature with webbing of the neck and lymphedema of her hands and feet. Intelligence may be impaired. Most affected embryos miscarry spontaneously. The most common deviation in males is Klinefelter syndrome or trisomy XXY. The affected male has poorly developed secondary sexual characteristics and small testes. He is infertile, usually tall and effeminate, and may be slow to learn. Down syndrome is an example of a trisomy. The cause of Down syndrome is one of three types of abnormal cell division involving chromosome 21. All three abnormalities result in extra genetic material from chromosome 21, which is responsible for the characteristic features and developmental problems of Down syndrome. The risk of having a baby with Down syndrome is about 1 in 1,250 for a woman at age 25, 1 in 1,000 at 30, 1 in 400 at 35, and 1 in 100 at age 40. Children with Down syndrome have characteristic features that are usually identified at birth. These common characteristics include small, low-set ears, hyperflexibility, muscle hypotonia, wide-spaced eyes, ulnar loop on the second digit, deep crease across the palm termed a simian crease, flat facial profile, small white crescent-shaped spots on the irises, open mouth with protruding tongue, and broad, short fingers. The outlook for children with Down syndrome is much brighter now than it was in years ago. Most children with Down syndrome have an intellectual disability in the mild to moderate range. With early intervention and special education, many learn to read and write and participate in diverse childhood activities. The principles of genetic disease inheritance of single gene disorders are the same principles that govern the inheritance of other traits such as eye and hair color. These are known as Mendel's laws of inheritance, named for the genetic work of Gregor Mendel, an Australian naturalist. These patterns occur due to a single gene being defective and are referred to as monogenic or sometimes Mendelian disorders. If the defect occurs on the autosome, the genetic disorder is termed autosomal. If the defect is on the X chromosome, the genetic disorder is termed X-linked. The defect also can be classified as dominant or recessive. Monogenic disorders include autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive patterns. Autosomal dominant inherited disorders occur when a single gene in the heterozygous state is capable of producing the phenotype. In other words, the abnormal or mutant gene overshadows the normal gene, and the individual will demonstrate signs and symptoms of the disorder. Autosomal recessive inheritance disorders occur when two copies of the mutant or abnormal gene in the homozygous state are necessary to produce the phenotype. In other words, two abnormal genes are needed for the individual to demonstrate signs and symptoms of the disorders. These disorders are generally less common than autosomal dominant disorders. X-linked inheritance disorders are those associated with altered genes present on the X chromosome. They differ from autosomal disorders. If a male inherits an X-linked altered gene, he will express the condition. Because a male only has one X chromosome, all the genes on his X chromosome will be expressed. The Y chromosome carries no abnormal alleles to compensate for the altered gene. Because females inherit two X chromosomes, they can be either heterozygous or homozygous for any allele. Therefore, X-linked disorders in females are expressed similarly to autosomal disorders. Most X-linked disorders demonstrate a recessive pattern of inheritance. Males are more affected than females. A male has only one X chromosome and all the genes on his X chromosomes will be expressed, whereas a female will usually need both X chromosomes to carry the disease. There is no male-to-male transmission since no X chromosome from the male is transmitted to male offspring, but any man who is affected will have carrier daughters. X-linked dominant inheritance is present if heterozygous female carriers demonstrate signs and symptoms of the disorder. All of the daughters and none of the sons of an affected male have the condition, while both male and female offspring of an affected woman have a 50% chance of inheriting and presenting with the condition. X-linked dominant disorders are rare. The most common is vitamin D resistant or rickets, a disorder involving a softening or weakening of the bones. Multifactorial inheritance disorders are thought to be caused by multiple genetic and environmental factors. Many of the common congenital malformations such as cleft lip, cleft palate, spina bifida, pyloric stenosis, club foot, developmental hip dysplasia, and cardiac defects are attributed to multifactorial inheritance. A combination of genes from both parents along with unknown environmental factors produces the trait or condition. An individual may inherit a predisposition to a particular anomaly or disease. The anomalies or diseases vary in severity and often a sex bias is present. For example, pyloric stenosis is seen more often in males, while developmental hip dysplasia is much more likely to occur in females. Multifactorial conditions tend to run in families, but the pattern of inheritance is not as predictable as with single gene disorders. Not all congenital disorders are inherited. Congenital means that the condition was present at birth. Some congenital malformations may be the result of teratogens. These are environmental substances or exposures that result in functional or structural disability. In contrast to other forms of developmental disabilities, disabilities caused by teratogens are in theory totally preventable. Known human teratogens are drugs and chemicals, infections, exposure to radiation, and certain maternal conditions such as diabetes and PKU. A teratogen has the greatest effect on the organs and parts of an embryo during its period of rapid differentiation. This occurs during the embryonic period, specifically from days 15 to 60. During the first 2 weeks of development, teratogens either have no effect on the embryo or have effects so severe that they cause miscarriage. Later on in the semester, we're going to go over some more teratogens, but a word that you may want to put in your memory is torch, T-O-R-C-H, infections. And these are different groups of viruses and things that causes malformations in our fetus. TORCH stands for toxoplasmosis, other infections, rubella, cytomegalovirus, and herpes. And like I said, we will be discussing these later on in the term. Viability refers to the capability of the fetus to survive outside the uterus. In the past, the earliest age at which fetal survival could be expected was 28 weeks after conception. With modern technology and advances in maternal and neonatal care, viability is now possible at 20 weeks after conception. Typically, we say that a baby is viable, which means it can live outside the uterus, typically between 22 to 24 weeks and a weight of 500 grams or more. The limitations on survival outside the uterus are based on central nervous system function and oxygenation capability of the lungs. The respiratory system begins development through an embryonic life and continues through fetal life and into childhood. The development of the respiratory tract begins in week 4 and continues through week 17 with formation of the larynx, trachea, bronchi, and lung buds. Between 16 and 24 weeks, the bronchi and terminal bronchioles enlarge and vascular structures and primitive alveoli are formed. Between 24 weeks and term birth, more alveoli form. Primary surfactant. The detection of the presence of pulmonary surfactants in amniotic fluid has been used to determine the degree of fetal lung maturity or the ability of the lungs to function after birth. Lecithin is the most critical alveolar surfactant required for postnatal lung expansion. It is detectable at approximately 21 weeks and increases in amount after week 24. Another pulmonary phospholipid, sphygmomyelin, remains constant in amount. Thus, the measure of lecithin in relation to sphygmomyelin, or the LS ratio, is used to determine fetal lung maturity. When the LS ratio reaches 2:1, the infant lungs are considered to be mature. This occurs at approximately 35 weeks. In order for us to do an LS ratio, we must do a amniocentesis on the mother, where we insert a needle into the uterus and pull out some of the amniotic fluid. This amniotic fluid is then sent to the lab and an LS ratio is determined. Now, typically your book says at 35 weeks the lungs tend to be considered mature. But in some pregnancies, such as my mom's who are gestational or diabetic, lung maturity takes a lot longer. In a mom with diabetes, at 35 weeks, the baby's lungs may not be mature. The fetal circulatory system, the cardiovascular system, is the first organ systems to function in the developing human. Typically between 6 and 8 weeks, we should be able to auscultate the baby's heart sounds. Fetal maturation continued. Hematopoiesis, or the formation of blood, occurs in the yolk sac, beginning in the 3rd week. Hematopoietic stem cells seed the fetal liver during the 5th week, and hematopoiesis begins there during the 6th week. The liver and biliary tract develop from the foregut during the 4th week of gestation. Coagulation factors 2, 7, 9, and 10 cannot be synthesized in the fetal liver because of the lack of vitamin K synthesis in the sterile fetal gut. This coagulation deficiency persists after birth for several days and is the rationale for the prophylactic administration of vitamin K to the newborn. The renal system. The kidneys form during the 5th week and begin to function approximately 4 weeks later. Urine is excreted into the amniotic fluid and forms a major part of the amniotic fluid volume. Oligohydramnios is indicative of renal dysfunction. Because the placenta acts as the organ of excretion and maintains fetal water and electrolyte balance, the fetus does not need functioning kidneys while in utero. However, at birth, the kidneys are required immediately for excretory and acid-base regulatory functions. The nervous system originates from the ectoderm during the 3rd week after fertilization. Purposeful movements of the fetus have been demonstrated in response to a firm touch transmitted through the mother's abdomen. Because the fetus can feel, the fetus requires anesthesia when invasive procedures are done. Bones and muscle develop from the mesoderm by the 4th week of embryonic development. At that time, the cardiac muscle is already beating. The mesoderm next to the neural tube forms the vertebral column and ribs. The parts of the vertebral column grow toward each other to enclose the developing spinal cord. Ossification or bone formation begins. If there is a defect in the bony fusion, various forms of spina bifida may occur. The epidermis begins as a single layer of cells derived from the ectoderm at 4 weeks. By the 7th week, there are 2 large layers of cells. The cells of the superficial layer are sloughed and become mixed with the sebaceous gland secretions to form the white, cheesy vernix caseosa, the material that protects the skin of the fetus. The vernix is thick at 24 weeks but becomes scant by term. The immunologic system. During the 3rd trimester, albumin and globulin are present in the fetus. The only immunoglobulin that crosses the placenta, IgG, provides passive acquired immunity to specific bacterial toxin. The fetus produces IgM by the end of the 1st trimester. This is produced in response to blood group antigens, gram-negative enteric organisms, and some viruses. IgA is not produced by the fetus. However, colostrum, the precursor to breast milk, contains large amounts of IgA and can provide passive immunity to the neonate who is breastfed. Endocrine. The thyroid gland develops along with structures in the head and neck during the 3rd and 4th weeks. The secretion of thyroxine begins during the 8th week. Maternal thyroxine does not really cross the placenta. Therefore, the fetus that does not produce thyroid hormones will be born with congenital hypothyroidism. If untreated, hypothyroidism can result in severe mental retardation. Screening for hypothyroidism is typically included in the testing when screening for PKU after birth. The reproductive system. Sex differentiation begins in the embryo during the 7th week. Female and male external genitalia are indistinguishable until after the 9th week. Distinguishing characteristics appear around the 9th week and are fully differentiated by the 12th week. When a Y chromosome is present, testes are formed. By the end of the embryonic period, testosterone is being secreted and causes formation of the male genitalia. By week 28, the testes begin descending into the scrotum. The female with 2 X chromosomes forms ovaries and female external genitalia by the 16th week. Oogenesis has been established. At birth, the ovaries contain the female's lifetime supply of ova. Most female hormone production is delayed until puberty. Multifetal pregnancy. The incidence of twinning is 1 in 43 pregnancies. There has been a steady rise in multiple births since 1973. This is partly attributed to delayed childbearing. The use of ovulation-enhancing drugs is also a factor. Dizygotic twins. When 2 mature ova are produced in one ovarian cycle, both have the potential to be fertilized by separate sperm. This results in 2 zygotes or dizygotic twins. There are always 2 amnions, 2 chorions, and 2 placentas that may be fused together. These dizygotic or fraternal twins may be the same sex or different sexes and are generally no more alike than siblings born at different times. Monozygotic twins. Identical or monozygotic twins develop from one fertilized ovum, which then divides. They are the same sex and have the same genotype. If division occurs soon after fertilization, 2 embryos, 2 amnions, 2 chorions, and 2 placentas that may be fused will be developed. Question. A woman is 8 months pregnant. She tells the nurse that she knows her baby listens to her, but her husband thinks she's imagining things. The most appropriate response by the nurse is A. Many women imagine what their baby is like. B. Babies in utero do respond to their mother's voice. C. You will need to ask the doctor if the baby can hear yet. Or D. Thinking that your baby hears will help you bond with the baby. And the correct answer is B. Babies in utero do respond to their mother's voice. Key points. Genetic disease affects people of socioeconomic levels and racial and ethnic backgrounds. Genetic disorders span every clinical specialty. Nurses with advanced preparation are assuming roles in genetic counseling. Genes are basic units of heredity responsible for human characteristics. 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Gestation is 280 days after the last menstrual period, or 266 days after conception, or 40 weeks, or 10 lunar months. Critical periods in human development during which embryo fetus is vulnerable to environmental teratogens, typically that first trimester, the first 8 weeks when organogenesis is taking place. This concludes the lecture on Chapter 8. Thank you.